Welcome to this first Oxford BRC Statistics Hub seminar on an introduction to clinical trials. My name is Jonathan Cook. I'm a professor of clinical trials and medical statistics based at the Nuffield Department of Orthopaedics, Rheumatology and Musculoskeletal Sciences at the University of Oxford. This is a short outline of the seminar that I'm going to go through today. I'm going to begin with a definition of a clinical trial. Then we're going to think about a history of clinical trials, followed by how modern clinical trials have evolved to the present day, followed lastly by a short summary. So, what is a clinical trial? There's been various definitions in use over the years. My personal definition is as follows. A clinical trial is a planned scientific study involving human participants which is conducted to learn about the safety and effect of a medical treatment or related care. And there's a few aspects of this I want to highlight. It's a planned scientific study. This helpfully differentiates a key feature of a clinical trial from other useful types of scientific studies. It involves human participants, so it excludes studies that are based on cells or laboratory-based or animal studies or other artificial scenarios that don't involve human beings. And it's a study that's looking at safety and effect of what? A medical treatment or related care. And why do we do these studies? Well, the objective might be put like this: to obtain an unbiased or fair and reliable evaluation of the treatment or related care of interest. Given that, when might we say the first clinical trial was conducted? Well, throughout history, we have medicines tested in various ways, mainly on informal basis, anecdotal and experience-based focus. Perhaps the first clinical trial in the proper sense, given the varying degrees of pre-planning that took place in these earlier studies, is the one by James Lind, who planned, conducted and reported on a study looking at 6 treatments for scurvy on 12 patients in the 18th century. James Lind was a Scottish naval surgeon, and these 6 treatments included options such as seawater, cider and vinegar, which had all been proposed for people, but it also included having oranges and lemons to consume. And those that received the oranges and lemons did better than the rest, and he wrote up his findings accordingly. And the thing is, it was a planned study. He conducted it, and he also reported the results. What we have from that time towards this day was steady progress, if not fast progress, towards what we might think of as a modern clinical trial. We have medicine solely reflecting the scientific approach, which had spread out from very narrow scientific experimental basis. And we have a gradual progression, particularly in the 19th century, towards a modern clinical trial where we see the use of quantitative measurements becoming more common in the medical area, the need for a reference or control becoming observed and noted, and the use of statistical data to summarize and even the beginnings of some sort of probabilistic analysis beginning. In many ways, the MRC streptomycin trial is seen as the epochal clinical trial, which was published in 1948. And I want to spend some time thinking about this study and why it was so important. The MRC streptomycin trial was a multi-center trial looking at streptomycin plus bed rest versus bed rest alone for the treatment of tuberculosis, and showed that streptomycin had a strong impact and reduced mortality in the short term. It was a study of only 109 patients at 7 sites. It actually started out with 3 sites and had to expand to 7 due to slow recruitment, so some things never change in clinical trials. 6 months follow-up. It had an observer blinded to make assessments about the recovery, and it was a randomized control trial. Though you don't find that term within the paper, it refers to random sampling numbers. What was really important, as much as the individual methodologies, it really came out of a system for delivering clinical trials. The MRC Therapeutic Trials Committee had been in place for a number of years, and therefore the learnings from previous studies fed into the conduct of this study. And it had very careful methodology, it thought through eligibility criteria. The randomization was applied in a stratified manner so that each site would be expected to be balanced, and it was using sealed envelopes to deliver those allocations. So an approach that's used even to the present day. There's trial-specific forms and instructions on assessment. There was investigator meetings, and there was a detailed analysis of results, including looking at subgroup analyses. In addition to the careful methodology, as important was the careful and clear reporting. This article was 18 pages, so substantially longer than many articles published today, and it included 4 pages of patient scans, so not something we would do in the modern day, but interesting to note. A key factor also was Bradford Hill, who was the main statistician involved in the MRC streptomycin trial and argued strongly for its use. He published 2 papers in the 1950s called The Clinical Trial, and this is the title section from one of those papers, which was published in the British Medical Bulletin, shown here, and you can see an outline of the topics which that paper covered. If we think about the modern clinical trials, I want to start off with thinking about the ethics involved. And we can see that these sort of considerations were part of the thinking behind the conduct of clinical trials and randomized controlled trials in the clinical setting from a very early stage. Indeed, that paper that I referred to had a section on ethics, though this was, to be fair, considered in a narrower sense than we would tend to do nowadays, thinking very much from the perspective of the doctor or the health professionals and their responsibility of care. Could they legitimately give the alternative treatments? Could they involve their patients in the study? There was, though, consideration of the role of informed consent, when it's appropriate to have a placebo control or have blinding. A very key moment was the watershed post-Second World War and the immediate response of the Nuremberg Code, which outlined important concepts and issues related to human experimentation. And we see some of these outlined here: voluntary consent and the importance of it, the right to withdraw from a study, the importance of risk minimization when you conduct a study, as well as gaining sufficient knowledge from the conduct of the study to justify the risks that are entailed. This was followed by the Declaration of Helsinki, which has actually been revised a number of times over the years to clarify, including how human experimentation is still possible and needed and acceptable, with guidance on when. Another key progression has been the concept of good clinical practice, that is how you implement your clinical trials, and there was a recognition for the need for an international standard, and was an international conference on harmonization held in the 1990s, and this has developed over the years and become legally incorporated for investigational medicinal products, that is a subset of drugs in the common language in the UK, the EU, the US and elsewhere. In parallel, and somewhat later, in its initiation, has been guidance and legislation for medical devices developed. If we think about those regulations a bit more, we can also note the impact of the thalidomide scandal and others, which led to laws in the 1960s and onwards mandating, and this is from the US law, substantial evidence through adequate and well-controlled studies of a drug's effectiveness prior to approval to distribute. This gave the FDA in the US the power to restrict the advertising and availability of drugs without meeting certain standards. And therefore a pathway developed. Though fluid, the overall target was consistent from the beginning, aligned with the law above, but regulatory requirements have grown massively since that initial law and this requirement initiated. Further legislation in the UK, EU and the US has occurred over the last 30 years for CTIMPs, that's clinical trials of investigational medicinal products, medical devices, and indeed are still continuing to be updated. And the expectation is further changes in the years ahead. All of this has led to some big distinctions in clinical trials. There's a difference in terms of expectations and documentation for those that are regulatory trials, that is those that are seeking, for example, approval of the FDA in the US, and non-regulatory, you might describe them as academic trials. In a similar manner, there's a big distinction between those which are drug, or at least IMP to be more precise, and to a lesser extent, device trials, at least where they're conducted under the regulations, against those that are non-drug trials. So other medical treatments don't, strictly speaking, have the same legal requirements for the evidence base that IMPs do. If we want to think about the design and statistics in the modern clinical, we can also see how they have progressed over the recent decades. The early trials predominantly focused on summary statistics and were graphically oriented. Statistical testing was sparse at the beginning, but the scientific, particularly statistical understanding, developed substantially in the 1970s to 1990s. We see considerations of sample size. Is our study going to be big enough to answer the question with any confidence that we're wanting to achieve? Perhaps we might be able to stop our study early, or should look earlier interim analyses, and if so, on what basis might we stop them, so-called stopping rules. We see a lot of development in the analysis methods in this period, and an increasing understanding of the need to focus on uncertainty, not just how unusual a result we have, or crudely put, just because we have a small p-value, that isn't the full story. And additionally, there's been recognition of the need to tailor and accommodate unique situations. Not all trials are the same, not all trials are conducted to answer the same question for the same situation, and the constraints do influence substantially what occurs in terms of the ethics, the practicality, and the time constraints. In terms of scope, the modern clinical trial, it's moved to all areas of healthcare and professions, from drugs to surgery to other treatments like CBT to acupuncture, etc. And in terms of range of scientific disciplines, we can take off economics, psychology, anthropology, regulatory specialisms, as well as the core trial delivery sciences, you might say, or skill sets of statistics, management, IT, and quality assurance have all had their influence and impact on clinical trials. And what we have now is an industry of clinical trials. That's a pharmaceutical company industry. We also have medical tech, which touches on clinical trials. We have institutes, whether charitable or governmental, funded that are involved in clinical trials, as well as universities and other types of organisations such as hospitals involved. We have centres of excellence. In the UK, we have clinical trials units, which specialise in the conduct of clinical trials, as well as clinical research organisations, or CROs, which specialise in the delivery of clinical trials. But what are modern clinical trials? What do they look like? Well, one way that people often think about clinical trials is the phases of clinical trials, and we might start with phase 1, moving 2, 3, 4 through, but each of those phases being slightly different in terms of focus and scale, starting from the smaller, perhaps only 10 people trying to answer the question, is it safe, or is there clear signal that we should not conduct any more human experimentation on this drug. It might be the first time that a drug or some other treatment has been used in a human. We're perhaps thinking about what's a safe dose, or at least are the immediate terrible side effects that might preclude progressing further. Often we're thinking about people who are getting treatment only, or healthy volunteers in this study. In phase 2, we move on to does it work? Perhaps we're thinking of 100 or 200 participants. Can we see some evidence of benefit? We're still thinking about safety. We might still be only looking at treated only, that is people who have received the new treatment. It might be a randomized trial, but perhaps not. And then moving to phase 3, perhaps the most famous of all the phases, where we're really looking for what's sometimes described as confirmatory evidence that this new thing does indeed work. We might be thinking of a scale of 1,000 patients. We are thinking of clear signs of practical benefit or real benefit demonstration. We still think about safety, but we do want to compare both against some standard, perhaps a placebo control or a routine care, whatever that might be. And most typically, we're talking about a randomized controlled trial. All being well, this might progress to a decision about approval in a regulatory setting. And then following that, a drug can be marketed and made available. And then we may get what's called a phase 4 study, where the drug is available to the public and being used on different types of people beyond what it was used on in the earlier studies, and therefore we're thinking about any adverse events that couldn't be anticipated from what went on previously, or perhaps as very rare side effects that we just did not pick up in the previous studies. Typically, though, we have no explicit reference here, and it's a clinical trial in a slightly different sense than these earlier studies. What do clinical trials look like in practice? Well, just to give a feel of how that slightly artificial framework doesn't apply by any means to every clinical trial, I'm here summarising the Chariot trial. This is one conducted by OCTRU, the Oxford Clinical Trials Research Unit, in collaboration with OCTO, the Oncology Clinical Trials Office, both based here in Oxford. It looked at safety, preliminary efficacy and tolerance of berzosertib with radiotherapy for treating esophageal and other solid cancers. And the first thing that you can see here is there's two cohorts or groups of patients, A1 and A2. So we've already deviated in quite a big way from that earlier simplified scenario where we've got two sets of patients and they have different criteria in terms of disease, the treatment that they're going to receive in terms of the duration or the schedule, And in this case, there was 16 in the first cohort when the study was finally completed, 20 in the second, so 36 people overall. This was a dose escalation study in the sense that it was looking at different dose levels, and rules for progressing were set, though it wasn't known at the outset how many participants would take part for certain and which dose levels would be received by a human participant. The areas of interest were toxicity, adverse events, and some indication of response, as illustrated in the diagram below. A very different study, again conducted by the OCTRU Trials Unit in collaboration this time with SITU, the Surgical Intervention Trial Unit, in particular the Orthopedics and Plastics Group, and it focused on 2 treatments for anterior cruciate ligament injuries, and the study was called ACL SNAP. This study had 316 participants conducted across 29 UK centres, and the main outcome of interest was the KOOS-4 at 18 months following entry into the study, and that's a measure patient-reported knee pain, symptoms and function. And then on this graph on the right, you can see how the KOOS-4 went up in both groups over time, but actually the surgical group performed better after 18 months. So you can see that clinical trials in reality differ from the artificial framework in many ways, because these differences and details are not legislated on, but are available to the person who's designing and conducting the study. And these practicalities shape the study, and it's worth highlighting some of these in particular. Typically, we're thinking of recruitment over time and slow, relatively slow, sadly, which means accruing data and follow-up information. The numbers of observations may also be limited. We may have diseases which even across the whole country, we might have very few individuals. For example, a rare pediatric cancer, for one case, only had 200 odd new cases in England across multiple years. So the type of study that you can conduct for that pediatric cancer is very different from looking at a very common medical condition. The data collection we can have is limited by practicalities. There's a limit to when we can collect data, realistically how we can get it from people. If it's patient reported, that has many other constraints about what is realistic to ask and what is realistic to receive from patients in terms of the quantity and the means that you do that. In addition, we need to collect often additional data beyond what's generally collected in medical care for the trial, but there's a limit to how much of that we could do. And so the efficiency of analysis is a key interest and concern in clinical trials. We have to deal here with complexity of data, which is another aspect from a statistical perspective. Clinical trials reveal events and situations that influence the observed outcome, and the importance of this can't be overemphasized. This is a great benefit of a clinical trial in that you understand the longitudinal process much better, and you understand and find out about things that are hidden in other simpler, what you might call flat data sources, such as routine ones like registries and healthcare system data. And so this facilitates a causal interpretation in that we are more able to say that this causes an adverse event or leads to an increase in that rate, particularly where we have a control and a randomized control ideally. So in summary, Clinical trials allow a robust assessment of the use of a treatment or change in care, but they vary greatly in their design, scale, and focus. Design and statistical approach is shaped by the practicalities, ethics associated with conducting clinical trials, which again varies very much so depending on the specific research question that's been asked, the treatment that's going to be received or treatments, where and how it's going to be conducted. I close this seminar with some references which I hope you will find helpful, or sources related to some of the studies that I have discussed in this seminar. Thank you for listening.